Okay, students, welcome to our next lecture about endosymbiosis and the origin of eukaryotes. You see pictures of different eukaryotes, including this funny monkey or ape, which is also a eukaryote, like you and me. I will just very briefly remind you what is the difference between prokaryotes and eukaryotes. Uh, the main difference is in the fact that the eukaryotic cells have membranous organelles. Usually people say the main difference is that eukaryotes have nucleus and prokaryotes do not. But this is correct. But the thing is that the nucleus surrounded by nuclear membrane is just one kind of membranous organelles. Others are endoplasmic reticulum, mitochondria, chloroplasts, Golgi complex, and other. And prokaryotic cells, as you see on this part of the slides, do not have these membranous organelles. Now, knowing this, we can go to the research that addressed the origin of eukaryotes. The research shows that prokaryotic cells were first on Earth. Right? Prokaryotic cells lived on this planet for a billion or more years. And then eukaryotic cells are thought to have evolved from prokaryotic cells. How was it possible? The first step in this evolution was hypothetically the infolding of the plasma membrane. See, here's an ancestral prokaryote. It has DNA inside. This DNA is not separated from the rest of the cell by the membrane. And then the plasma membrane makes an infolding, right? And the result of this multiple unfoldings is that you see special formations inside the cell which are membranous organelles, endoplasmic reticulum, nucleus, cell with endomembrane system. It is, and the nuclear envelope is also, it's a membrane actually. So that is the evolution from prokaryotes to eukaryotes. Then this is the summary again of this hypothesis of endosymbiosis. And this is graphically very nicely shown. First cells were prokaryotic. Organelles like mitochondria and chloroplasts were independently living cells similar to bacteria. You see this green bacteria. It is a prokaryotic cell, and it has chloroplasts which produce this green pigment chlorophyll. And then the cells that engulfed mitochondria or chloroplasts, on this picture is clearly chloroplasts, received some evolutionary advantage. For example, cells that engulfed chloroplasts used them for photosynthesis. You've heard the term photosynthesis, extracting energy directly from the sunlight. And mitochondria for breaking down nutrients and producing ATP. So cells that became eukaryotic became kind of favored by evolution. They multiplied greatly. Although prokaryotic cells still exist. We know that bacteria exist, archaea exist. They are prokaryotic. But eukaryotic cells kind of took over from approximately 1 or 1.5 billion after the appearance of prokaryotic cells. This is some explanation of this hypothesis in somewhat more detail. See, the picture shows endosymbiosis that helped to diversify the eukaryotes into animals and plants. The animals and plants are both eukaryotes. And again, there was this ancestral prokaryote. See, it engulfed some bacteria which were aerobic, meaning they could use oxygen uh, to, to produce, to generate energy. And those cells that engulfed these free-living aerobic bacteria became cells with mitochondria, but without chloroplasts. Which cells have mitochondria but not chloroplasts? Animal cells, right? Then uh, other cells engulfed some bacteria which had pigment in them, like cyanobacteria. They have green pigment which allows photosynthesis to occur. So they became plant cells. These cells, they became autotrophs. They can feed themselves by extracting energy from the sun. So endosymbiosis helped not only to appear, but also it helped to diversify eukaryotes into plants and animals. And pro and eukaryotes form the tree of life. See, there is a there exists two domains which are prokaryotic: bacteria and archaea. And due to endosymbiosis engulfing either chloroplasts or chloroplasts and mitochondria, cells became eukaryotic and diversified into plants or animals. Or here you have a special group, fungi, mushrooms. So these different groups of living creatures are called kingdoms. So there are three kingdoms. There are protista, cells that sort of are still thinking about what to become, plants or animals. Then there are plantae, Latin plants in English, and animalia, animals in English. Protists, protista, they are the earliest kingdom. They were first eukaryotes. It's the starting point of evolution of fungi, plants and animals. Let's take a look at protists. So what are they? Uh, they are a huge kingdom. There are so many representatives of protists, and they're very diverse. They are defined by exclusion. Right? If you see a cell and it is eukaryotic, but you cannot classify it as cell of a plant or cell of an animal or cell of a fungus. So you say, yeah, that's protists, right? Usually they are unicellular, and although some of them are colonial or multicellular, and they can be plant-like or animal-like, or it's not shown, not seen on the slide, but also fungus-like, mushroom-like. See here are examples: animal-like, top left corner, animal-like uh, creature called chaos diffuens. It's basically an infusorium. It is a cell that has cilia on it, little appendages which move all the time and allow the cell to move. And note, it has no cell wall, only cellular membrane. It has mitochondria but does not have chloroplasts, so it cannot do photosynthesis. Then there are plant-like protists. There's Eubodiscus radiatus. It's a cell that moves due to the flagella, long appendage that works like a propeller in airplanes. And you see, it's covered, it's colored green, and in nature, really, it is green because it has chloroplasts, which produce a lot of chlorophyll, so it makes it look like plant-like cell. Still, neither these chaos diffuens nor the Eupodiscus radiatus, you cannot classify them as animals or plants. They are protists, of which one is animal-like and the other is plant-like. Now, there is also a subkingdom of fungus-like protista. Here's the example is Fuligo septica. It's because septica is probably pathogenic for for humans. It does not have a lot of pigment. Actually, it is colored yellow, but yellow pigments do not participate in photosynthesis. So it cannot be a plant. And it is not also an animal because it doesn't move, it's sedentary, and it does not feed the way animal cells feed. Animal cells feed by engulfing something. But this cell has little threads, fine meshwork of threads, which are called hypha. And these hypha, they are like absorbents. They drag nutrients from the surroundings of this cell. That's the typical way of feeding of fungus. But again, in the cell, this is a fungus-like protist. It's not real fungus. And finally, you see here's an example, not frequent example, of multicellular protists. Seaweed, Fucus vesiculosus. You don't have to memorize these names. Many cells, right? Many cells. So protists are the earliest kingdom where cells are neither plants nor animals nor fungi, but can display features of plants or animals or fungi. And it is also the kingdom where multicellularity appears. And you first see the forms of life that consist of more than one cell. Organism is more than one cell. Let's take a look at animal-like protists. They're also called protozoans. Zoa also refers to animals. The zoe means life, but traditionally zoa it's a reference to animals. There's a cell that is called, it's also an organism, one cell, one organism, called paramecium. And this is the picture photo, and this is the diagram which shows you the organelles, membranous organelles of this paramecium. So it is definitely a protist, and it is definitely a eukaryotic cell. It has large nucleus surrounded by a nuclear membrane. It has contractile vacuole. Note that this vacuole is relatively small. In plant cells, you have huge vacuoles, and they're not contractile. This is a contractile vacuole. When it contracts, it pushes the cell to some direction. And then there is a cell membrane, but no cell wall, right? So animal-like creature, paramecium. And here there's another animal-like protist, also unicellular. It's called Giardia, full Latin name Giardia lamblia. And here is one very strange and terribly nasty protozoan called Plasmodium. These red balls, they are red blood cells, and Plasmodium is this tiny little yellow ball which invades into a human red blood cell and destroys it. So some protozoans, like Paramecium or that infusorium that you saw in the previous picture, they are harmless. Other protozoans are dangerous pathogens. Giardia causes a severe diarrhea, and Giardia is usually the cause of water-induced diarrhea. When you travel outside of the United States, Mexico is notorious for having defects in the filtering systems, and the tap water in Mexico, I have a personal experience in that, has tons of giardias. And so if you travel to Mexico, my advice, stay away from giardia, drink bottled water only, not tap water. And Plasmodium, the tiny ball that invades the red blood cell, it causes malaria. Malaria is a very dangerous disease. It manifests as very high fever that lasts either 3 or 4 days, then a relapse, and then again fever, and then again relapse, etc. It's because the red blood cells damaged by Plasmodium are then they travel to the liver and cause inflammation of the liver with these waves of uh, high temperature fever. Plasmodium is a killer. People who have malaria, very many of them die, and the appearance of anti-plasmodium medicines actually played a very, very beneficial role in increasing the population of countries like sub-Saharan Africa, Pakistan, India, etc. And then, next slide, animal-like protists, amoebas, OE, pronounced E, so amoebas. Amoebas, here I made them a separate slide because they are very interesting. They are like chameleons. They can change the shape of their bodies. So an amoeba can be rounded, and then it grows appendages and becomes like this one shown in the right upper right corner. It can squeeze between cells and between other objects. The reason why they can change their shape and also the reason they can move and feed is that they grow appendages called pseudopodia. Literally, pseudopodia means false feet. It's not feet, of course, but those are appendages that are temporary extensions of the cell bodies. They form because amoebas can relocate the cytoplasm toward one side of its body and make the side of the body form kind of like extension, temporary extension. When amoeba feeds, it grows pseudopodia and then embraces its prey. It's a predator, actually. Embraces its prey with these pseudopodia and swallows it. The prey is usually bacteria, or they can be other protists, smaller, defenseless. Amoeba doesn't really care. Now, why is this interesting? Because in upper organisms and animals and humans also, there are special white blood cells called phagocytes. They perform so-called phagocytosis, and this is exactly the way amoebas engulf their prey. These phagocytes grow pseudopodia, surround or embrace a bacterium, swallow this bacterium, digest it, and use the resulting nutrients for generation of energy. So phagocytes are very beneficial. They are a tremendously important part of the immune system in humans because they swallow bacteria, including very pathogenic, nasty, disease-causing bacteria. Fungus-like protists, slime molds, as interesting creatures. They resemble fungi by appearance. See, this looks like sort of like mold or something. And by the mode of feeding, they are decomposers, so they also absorb nutrients from their environment, like suck them in with the help of hypha, and then decompose them, digest them. And interestingly, slime molds can grow to a rather large size. See, there's a human being here, and this is a slime mold, so it's huge. This slime mold also pretty big, but always they are unicellular. Right? So this big blob of some slime, I don't know, it's actually one cell with many nuclei. They are multinucleated. And in this fungus-like protist slime mold also, this is one cell with many appendages and many nuclei. It's interesting sort of twist of evolution. And plant-like protists. Algae, this is pronounced algae. Uh, Algae, I'm not sure whether it's right pronunciation. It's understandable, but the correct one is algae. Please remember that we say fungi and not fungi. we say algae and not algae. Algae are unicellular or colonial. Here in the lower right corner, you see colonies of algae, uh, or multicellular. They can be multicellular. Uh, their cells resemble plant cells. Why? Because they have cell walls, and they have chlorophyll, and they perform photosynthesis. Uh, the chlorophyll-like pigments allow them to harness energy directly from the sun. That's what the essence of photosynthesis is. So they do not need nutrients. If they are swimming in water, they get their energy from the sun. They don't need to they don't need any food. Most of them are green, but some of them have red pigments, which some of these red pigments also can perform photosynthesis. Uh, One nasty thing about these so-called red tide or red colonies of algae is that if the storm or waves move these colonies of red algae toward the beach, they disintegrate on the sand or on the rocks of the beach, and the products of their disintegration are toxic for humans. So red algae are not friendly. Uh, then multi. Oh, that's another interesting kind of unicellular plant-like algae. Uh, they are unique in that, see, their cell walls are very uh, thick and rigid because in their cell walls they have large amounts of substance called silica, and this is a substance found in glass. So they are these tiny little diatoms. They're really resilient to pressure. They're like tiny pieces of glass, nonetheless living cells. A powder prepared from diatoms called diatomaceous earth is used as insecticide. It kills insects. And also the medium to bury radioactive isotopes. It's very important to use. I don't think your textbook mentions it, but I personally worked with radioactive isotopes in laboratory, and I know that it's a big problem to get rid of them. You cannot dispose radioactive isotopes in a sink or in a trash can because they are radiating. They're dangerous. So scientists who perform experiments with radioactive isotopes buy sacks of diatomaceous earth, and they dispense or dispose radioactive isotopes into this diatomaceous earth. Radiation hardly can even reach outside of the diatomaceous earth. And then these sacks of diatomaceous earth are picked up by special service and buried in a secret place which only these teams that do the burial know. And these, of course, these burial places are regularly inspected until there is confidence that radioactive isotopes are no longer dangerous. Uh, Finally, about algae, there are some multicellular algae which we call them seaweeds. They may be the first truly multicellular form of life, and they grow to gigantic size. So their cells are capable of division at a very high rate, and these seaweeds from bottom to top may be many hundreds of yards tall. You might recall the geographic name Sargasso Sea. It's the sea in the Caribbean region. It is so full of colossal seaweeds that it's difficult for a ship to proceed because the seaweeds are interfering with the engine. It's multicellular algae for you. Interesting point, evolution of multicellularity. Why did cells join and remain together? What advantage does it confer? And again, the research shows that the earliest living organisms were single-celled. Then they formed colonies at some point. Staying in a colony is advantageous because it is a colony is better protected from predators, right? Remember this amoeba swallowing a bacterium. And this cell is small size, kind of like bacterium. So an amoeba would swallow it in an instant. But this colony, oh, that's a different thing. The amoeba is helpless. It will not swallow it. Then at some point when these cells in a colony replicated, cells that happened to be inside the colony died because they do not have enough nutrients. Nutrients are absorbed from the environment by engulfing something, etc. The cells inside this huge blob are not receiving enough nutrients, so they die. And hollow spheres formed. So an important step in the forming of multicellularity is when there is a superficial layer or layers of cells and a cavity, an empty space inside. You know, you have cavities, right? You have chest cavity, you have abdominal cavity. So that's evolution for you. The organisms evolved from these blobs of cells to structures that have cavities or hollow spheres. Then there occurred some differentiation between the cells that surround the hollow sphere. Some of these cells had faster way of dividing, higher mitotic index. So they became reproductive cells. Other cells had better ability to engulf bacteria or to absorb nutrients from water, etc. So they did not differentiate into reproductive cells. And finally, at some point, tissues appeared because cells further differentiated into very superficial, which ectoderm, that's the skin, the covering tissue, middle, which is like connective tissue, and specialized cells like cells that are reserved specifically for reproduction. Okay, next we are kind of making a slight turn and talking about viruses. See, when we remember this tree of life, it said that there are bacteria, archaea, protists, plants, fungi, animals, right? It did not include viruses. We hear this term virus. Virus causes influenza, virus causes this, virus causes that. So why are viruses not included into the tree of life? Viruses are very difficult to define, but one thing is clear. Outside of the cells that they infect, they're not living. They don't show any features of life. No biochemical reaction proceeds inside the virus, right? No energy is generated, neither by photosynthesis nor by absorbing and splitting nutrients. No metabolism, completely inert structures. So outside of the cell, viruses resemble little grains of, I don't know, rock or salt or something. They're like mineral substances, not living, definitely not living. So that's why they're not included in the tree of life. And please do not confuse them with bacteria. I personally cringe when I hear or read that students say that this bacterium is a virus. No, bacteria are bacteria, viruses are viruses. They're separate entities. But what is characteristic for viruses? They can infect cells, so invade into a cell. And when they invade or infect a cell, they force the cells to replicate them. Viruses have genetic material, either DNA or RNA. They cannot use it for replication because replication requires metabolism, energy, etc. Viruses do not have that. So they're parasites. They use the energy of the cell, the metabolism of the cell, the enzymes of the cell, all the reproductive machinery that services the copying of their genetic material. And in case of severe virus infection, cells become totally unable to replicate their genetic material. They turn into factories that produce little baby viruses, replicating the viral DNA or RNA. Viruses may kill cells. Some viruses are cytopathic, or cytotoxic, or cell killing. If there's a virus that invades the cell, then this virus produces progeny using the cellular machinery, and it can be hundreds or thousands of little baby viruses. And then these little baby viruses sort of mature and become adults, and they rip the cell entirely. They destroy the cell, and they are out of the cell and ready to infect another cell. So that's how viral infections spread. Generic sort of structure of viruses. Viruses have either DNA or RNA, never both, right, inside. So this virus apparently is a DNA virus. It's the double spiral. Then this genetic material, DNA or RNA, is surrounded by a structure called capsid. It's mostly proteins. And this capsid may or may not be further surrounded by envelope. Envelope is lipid or fat by origin, and viruses do not produce it. Viruses do not make it. They steal it from cells that they are infecting. Some viruses do not have envelope. They're called naked viruses. Others have envelope and called enveloped viruses. Some particular viruses, influenza virus. It's an RNA virus. It's interesting that it has fragmented genome. It doesn't have one piece of RNA. It has multiple pieces of RNA, which allows these viruses to exchange these little pieces of RNA. And this exchange may generate viruses with very unusual features, like this swine flu, bird flu, etc. There is a very limited number of kinds of cells that influenza virus can infect. Cells of the respiratory tract. Viruses infect them and kill them, right? So that's why people who are sick with flu, usually the beginning of the flu is symptoms of intoxication, because millions, literally, of cells of the respiratory tract are dying and spilling their guts, which may be toxic for the organism, and you feel fatigue, you generate high fever, you feel pain in the muscles. Muscle aches are very characteristic symptoms of influenza. Then headache and shortness of breath, cough and nausea. Please note that influenza viruses never infect cells of the gastrointestinal tract. So when people say, I have a stomach flu, they are wrong, because it's not influenza that causes this array of symptoms in the large and small intestine. It's maybe adenovirus or some other virus, but not influenza. Okay, other examples, herpes virus. Herpes is a DNA virus. It infects the skin. And there are two species of it, herpes virus 1 and herpes virus 2. Herpes virus 1 infects skin above the waist. So most common manifestation is cold sore. You see this upper picture is cold sore. The other herpes virus 2 causes genital herpes. It infects cells on the genitalia. Now, the herpes virus, for many years, it was a suspect that was thought to cause cervical cancer in women. Actually, it does not. It's one of the several strains of papillomavirus. Several strains of papillomaviruses cause cervical cancer. Still, genital herpes is dangerous. In women, especially, genital herpes may lead to infertility. And one nasty thing about genital herpes is that most people infected with genital herpes do not know that they have it. Some studies suggest that approximately 20 or even more percent, or so every fifth person in the United States has genital herpes, but very few of victims of this infection realize that they have it, and they spread it to their sexual partners. So it's not a good thing. Then human immunodeficiency virus, HIV. That's really a very nasty kind of virus. It is called retrovirus. Why retrovirus? Tro is a sort of a little bit distorted transcription. Remember from Bio1 that the genetic material is transcribed and then translated, and the transcription goes from DNA to RNA. In retroviruses, it's the other way around. Their genetic material is RNA, but they have special enzymes that help replicate this RNA and transcribe it into DNA. Why is it bad? Because this viral DNA inserts itself in human genome, in human chromosomes, and behaves like a gene, although of course it's not a human gene, it's a viral material. So people who are infected with HIV develop very severe infection. The target of HIV, target number 1 is cells of the immune system, T helper cells, dendritic cells, macrophages. So the immune system suffers. That's why it's called immunodeficiency virus. The immune system is deregulated. And what it means is that we all have some opportunistic infections. So we have some infectious agents that live in us and do not even let us know that we have them. Like there is a yeast unicellular fungus called Pneumocystis carinea. Almost all people have it in the lungs. It lives between the alveoli of the lung. But when the immune system is normal, Pneumocystis carinea is under check. The immune system does not allow it to replicate, reproduce to very large numbers. But when HIV destroys the immune system, Pneumocystis carinea begins to replicate with fantastic speed, and very many people who have AIDS actually die because of this virally induced destruction of the lung. In some severe AIDS patients, lungs simply do not exist. They actually are sacs filled with Pneumocystis carinea. So there is no respiration as such, and these people must be on the ventilator and the artificial life support. Or also tumors. The immune system normally keeps tumors under check, but again, when it is destroyed by HIV, tumors grow. This is probably the most common tumor in AIDS patients is Kaposi's sarcoma. It's the infection of the muscle cells, and it manifests as lesions on the face. That's because of the tumors growing in the muscles that are participating in mimics in smiling, frowning, etc. Then in the anal area also there might be a huge number of these tumors which actually they make a person incontinent, etc., etc. Then just a little bit about, I know this is not a virology course, but still, since we're talking about HIV, how do people get HIV? There are three ways of getting HIV. One is unprotected sexual intercourse with an infected partner. Another is vertical transmission, meaning mom gives the virus to her fetus, and then the baby is born already infected with HIV. And finally, injection with usually shared needles. Some drug addicts, I don't know, it's kind of crazy, but they like to share needles. One syringe with one needle goes into veins of different people. That's an extremely risky thing to do, because that actually you may contract virus when you, or HIV virus when you do that. How to prevent HIV infection and AIDS? First of all, live simple, godly life. Do not engage in sex with God knows how many partners, especially unprotected unprotected sex. Then, if you are infected, people are screened for HIV. For example, when you donate blood, your blood will be necessarily tested for HIV. And if you are found positive, God forbid, you have to take antiviral drugs. It's a must, because if you don't do that, you become an incubator for the virus, and you spread it around you when you engage in sex and when you are sharing needles or something. And you risk to shorten your own life. So yes, get this cocktail of anti-HIV drugs. They are available in any medical office, and it's just a must. Then some newer kinds of viruses. Ebola, you probably heard about Ebola infections. Right? Ebola is a spiral-shaped virus. It's an RNA virus, and it spreads very quickly in the human organism. Major damage to the human is impairment of blood clotting. Basically, the virus causes blood to clot inside blood vessels. So basically, the circulation stops, and a person infected with Ebola virus usually is dead after just several hours. And in sub-Saharan Africa, again, that's the birthplace of Ebola virus, and that's where there's this huge reservoir of Ebola viruses. And epidemics arise every now and then, and whole villages, whole living areas can be depopulated. Not a single living person remains. It's a very dangerous virus, and a lot of people are working on vaccine against Ebola. So far, not very successful, and no one really knows how to prevent Ebola, because when the epidemic arises, this virus is spread all over. It is spread by sneezing, coughing, spitting, defecating, urinating. Everything. The person who is infected really spreads the virus all around, and it's very dangerous. Sorry for the barking dog. Okay, also here in Mississippi, one kind of virus that is pretty nasty. It's West Nile virus. It's a virus that is transmitted by mosquitoes. Right? So extermination of mosquitoes is extremely important. It can cause skin rash, usual symptom of West Nile, high fever, and most dangerously meningitis, the inflammation of the connective tissue that surrounds the brain, and it can be lethal. So prevent yourself from West Nile. If you have some bodies of water in your backyard, liquidate them. They are the breeding ground for mosquitoes. Also use anti-mosquito sprays, etc. And whenever possible, if you hear about an, uh, an outburst of Nile virus infection, protect your body from mosquitoes. Wear long pants, wear long skirts or something, and do not allow mosquitoes to bite you. And the last example of viruses, measles, mumps, rubella. All these three diseases are caused by viruses, three different viruses, all of them RNA. Each of them causes skin rash because it targets small blood vessels in the skin, destroying them and causing this characteristic skin rash. Mumps virus infects cells of the parotid salivary glands, which are located under the ear lobes. So the face of a child, usually children before the age of 5, 6, get mumps. It becomes sort of like a face of swollen face. From parents may be quite afraid. Why the swelling? What's going on, etc. It's not a severe disease. Measles, on the left, it is a severe disease. It may spread on lungs and cause lethal pneumonia. May spread on brain and cause meningoencephalitis, also lethal. Mumps is not as dangerous, but there is one complication. In boys, there is another target of mumps virus, and other besides the parotid salivary glands, testis. And when mumps virus replicates in the testis, it messes up the structure of the testis, and it can cause male infertility for life. It's not often, but it happens. So, vaccinate your children, vaccinate your children, vaccinate your children. Vaccination does help. Vaccines do lower the incidence of diseases like measles, mumps, and rubella. So please, please, please vaccinate your children. Don't listen to people who say that vaccines are themselves disease. They're not disease. That vaccines cause lethal complications. Yes, they do, but they cause these lethal infections in like one person out of million, while thousands of people, when they are vaccinated, manage to evade the disease. So do vaccinate your children. Then making another turn now back to the tree of life. Fungi. Remember, on that tree of life, there was a diversification into plants to the left, animals to the right, and fungi in the middle. A very common very numerous kingdom of life, kingdom of eukaryotes. And it includes multicellular organisms like these mushrooms, and it includes also unicellular organisms like the yeast, which makes this dough rays, right? Yeasts are unicellular fungi, so variety of forms. What unites all fungi is that they are decomposers. They feed by breaking down large organic molecules of other organisms. Right? As you already know that uh, fungi drag some juices from their environment using their hyphae, and um, very often fungi grow near or on dead bodies, so they help decompose these dead bodies of animals, etc. So that they are decomposers, and this is kind of like a black humor joke on the right that before 1791 Mozart was composing, and since 1791 he's decomposing. Remember, this is actually really kind of black humor, but remember that this decomposition of dead bodies, to a very large extent, it's the useful work of fungi. There are other decomposers, but fungi are probably the most active. The other types of fungi, yeasts, unicellular fungi, then molds, which can be unicellular or multicellular, and mushrooms that are always necessarily multicellular. What is also common for fungi, uh, many of them are used in human activity. They do useful things. For example, yeasts are fermenters. Fermentation is a process when carbohydrates are broken down, but not completely. So not very large amount of energy is gained through fermentation. But these products of incomplete breakdown carbohydrates may include little bubbles of air, which cause the dough to rise, acids like lactic acid, which is the reason why we have sour cream, yogurt, etc., etc. Some yeasts also produce ethyl alcohol, which is they are used in beer making and wine making. They were discovered by probably ancient Egyptians thousands and thousands of years ago, and they're still used in alcoholic beverage industry. Then molds. Molds are predominantly multicellular fungi. Some molds, like this shown here, produce antibiotics, substances that kill bacteria. There's a picture of Sir Alexander Fleming, who was the person who discovered penicillin. And it was an amazing discovery because what happened in Fleming's laboratory, some bacterial cultures were contaminated with mold. And probably 99 people out of 100 would just toss away these contaminated cultures and forget about them. But Fleming examined them, and he noticed that when there is a colony of fungi, around this colony there is a clear agar with no bacteria. Right? So mold produces a substance that kills bacteria, because the species name of that mold was Penicillium notatum. Fleming called the substance penicillin, and it is still used in pharmacological industry, and there are dozens of derivatives or substances that are similar to penicillin, like carbenicillin, ampicillin, etc., etc. They may be quite efficient against bacteria. Unfortunately, in some people, they cause allergic reactions, sometimes may be very severe, and then they cannot be used. Then mushrooms. Mushrooms are multicellular fungi. They have mycelium, which is like a huge network of hyphae, these threads that can absorb nutrients. They also absorb water. And you see there are there are pictures of mushrooms that can be eaten and mushrooms that should not be eaten under any circumstances. There are some toxic mushrooms. This picture on the right, it's a mushroom that produces substances that are similar to opioids and also hallucinogens. So it causes very nasty intoxication and um, temporary insanity by hallucinations, etc. Now, fungi are the first organisms in evolution who started to reproduce sexually, right? How is it possible? The thing is that the hypha in mycelium, the sum total of hypha, they are, we say, haploid. So they have a certain number of chromosomes, say 30. I'm not sure exactly what. Let's imagine that 30 chromosomes. Then mycelium of one fungus can fuse with mycelium of another fungus. So hypha can fuse together. And thus, a zygote is formed. Zygote is a diploid cell, meaning it gets 30 chromosomes from one partner and 30 chromosomes from another partner. So total number is now 60. And then the zygote undergoes a process which you should know very well from biology 2, from biology 1, meiosis. Meiosis. It's a process where the pairs of homologous chromosomes separate and end up in different gametes, spores. Right? And spores again are haploid, and they can reproduce mitotically, forming new hypha, and then again hypha may fuse together, forming diploid cells which undergo meiosis. Why is that important? Because see, when organisms replicate asexually, for example, bacteria, when they replicate, they just split into two cells. And then these two cells form 4 cells, then 8, then 16. All these daughter cells are exact copies of the parental cell. So this population of bacteria is actually a clone. And there's very little genetic diversity. Of course, bacteria mutate, like all organisms mutate. So there is some diversity in a population of bacteria. But in a population of organisms that reproduce sexually, because of the meiosis, which, remember, it includes this independent assortment of chromosomes into male and female gametes, it introduces a huge genetic diversity, which is beneficial for the population because it allows the population to withstand calamities. For example, imagine that the acid rain changed the pH of the soil where these mushrooms or other kinds of fungi grow. That if all cells were genetically identical, if all organisms were genetically identical, they would probably all die, right? Low pH is not conducive to living. But if meiosis occurs, there might be some combination of maternal and paternal chromosomes, which confers resistance to pH changes. So sexual reproduction is very beneficial. It's like an insurance policy for a population of organisms, which protects them, makes them live longer and reproduce to larger numbers. Now, there are some parasitic fungi. See the ringworm. It's not a worm. Remember, when somebody tells you that I have ringworm, don't think of worms. It's a fungal infection which is usually on the hands and feet, but it may be other locations too, and it is pretty nasty. It is itchy, and when people scratch these itchy colonies of ringworm, they spread them, so they sort of help the fungus to replicate in their organism. So they should not do it, and there are, thank God, and scientists, there are some anti-fungal creams, ointments, etc., and also pills and injections of antifungal medicines. Then athlete's foot, right, upper right corner, very common fungal infection. Usually people get it when they share shower cabins, and these shower cabins usually have a wooden sort of platform, which it's not feasible to change it into another one after every person who showers. So it stays there, and if someone has athlete's foot, stepping on this wooden platform will bring fungus to this platform and may infect other people. Same thing as ringworm, very itchy but treatable with antifungal medicines. And pneumocystis, we already mentioned it when we talked about AIDS. It's a yeast, single cellular fungus, which lives in the lungs, and when the immune system is destroyed, it destroys the lung. And it's probably #1 cause of death in AIDS patients, although some claim that lymphomas are #1 and pneumocystis is #2. Okay, now fungi in ecology, interesting point. Fungi are decomposers, right? So in the world of living organisms, there are producers. Plants are producers, like green algae are producers. All organisms that run reactions of photosynthesis, they all produce organic molecules, including nutrients, which are important for their life, and also for the life of the creatures that consume them. This little mouse that eats grass, so it eats the producer. It's called primary consumer. Then, see, there is a snake, third from the left. The snake can swallow the mouse, right? So it's a secondary consumer. Now, and there is a falcon, which easily can attack this snake from the altitude, grab it into its feet with talons, etc. And it eats the secondary consumer, so it becomes tertiary consumer. But there are also decomposers. Fungi are decomposers. Why they are so important in the food chains? Because they support producers. #1 benefit from having decomposers is that they're natural fertilizers. See, a grass can grow without any nutrients. Even shrubs and bushes and even trees can grow for a while without any nutrients, just due to photosynthesis. But if the soil is fertilized, decomposers actually fertilize the soil, then plants grow better, grass grows faster, trees grow taller, etc. So it positively influences this entire food chain. So that's why fungi are very important. And we will stop here, and the next lecture will be on other eukaryotic organisms, but this slide will cover the lecture on the origin of life, the lecture on the diversity of prokaryotes, and this lecture on eukaryotes, their origin and their diversity. Thank you guys for listening. Best wishes for the next quiz and generally for the course.